Hey guys, happy Friday. Maybe not so happy. If you're like me, you just found out that we're not going back to school at all. I'm pretty disappointed about it. Maybe you're not, I don't know, but I expect you probably are, especially seniors. Be nice to see everybody again, nice to do a proper graduation and everything, but I don't know, we'll see what happens, but it doesn't seem like any of that's going to happen. So anyway, let's get into this second PowerPoint here. There's a lot of questions in this one, so I'll let you know when you should know the answer, and you should take some time to think of it on your own. So the monomers of nucleic acids, and I'm not sure if you remember this is going back to freshman bio, but they're called nucleotides. And nucleotides are made up of three basic parts. And I don't know if you remember those, but there's a phosphate group. There's a sugar that is a 5-carbon sugar, 1, 2, 3, 4, 5. That's why it's called a pentose. And then there's a nitrogenous base, and those, of course, come in 4 different flavors: adenine, thymine, cytosine, guanine. That pentose sugar, if you look right there, you see that oxygen on this molecule. That oxygen is missing, so this is ribose. And if I take away one of the oxygens, then I have deoxyribose. I've taken off one of the oxygen, deoxy. So that's where DNA gets its name, its first name anyway, deoxyribo. The double helix structure, of course. We kind of looked at this briefly last semester, but I don't expect you to remember. But the DNA is, right, it's set up with each of these. So that's a nucleotide right there. There's another one right here. And they're connected. Each phosphate is connected to the previous sugar. So, and if you look on this side, we've got the same thing, but it's actually running the opposite direction. It looks like it's upside down. DNA is what we call antiparallel, which means they're parallel, but running in opposite directions. We'll talk about the direction stuff later on. Carbons on any organic molecule can be numbered. There's a whole system for this. But when you've got a ring structure, like you do with ribose here, the carbons, you start counting the carbons, or you can see there's, here, I'll circle each of these carbons. Remember, any intersection, when we're drawing organic molecules, we just put these little intersections. That means there's a carbon at those points, unless it is already labeled with another letter, which there's an oxygen here at the top. So I've got 5 carbons here. 1, 2, 3, 4, 5. And one is off of the ring and the other 4 are on the ring. So by convention, what we do when we number these is we start with the one that's on the ring if you have this situation. So this is the number 1 carbon of ribose, the 1' carbon. So we have a 2' carbon, a 3' carbon, a 4' carbon, and a 5' carbon. And this will come in handy here in a bit. We'll need to know about this 3' and this 5'. Those are the carbons that connect it all together. As this molecule is built, right, so here's that phosphate again, and it's attached, right? So the phosphate is attached to the 5' carbon. That is attached to the 3' carbon of the next nucleotide, or the next monomer of DNA or RNA. So you end up having, as you chain more of these together, you end up having a 5' end, right? So there's this end with the 5' that's not attached to another 3' up here, right? That's not there. So you have a 5' end, and then you also have a 3' end. So the molecule itself, that chain, has 2 different ends. And that's always the case with polymers. We just don't always mention it. So these strands are anti-parallel. Like I said, they run in opposite directions. I've got the 5' end at one here and the 3' here. And on the opposite side of on the other side of each of these base pairs, that strand is running in the opposite direction. So it's 5' to 3' running the other direction. So again, anti-parallel, parallel to each other, but running in opposite directions. And that's what we mean by that. So we have a 5' end, we have a 3' end. That'll be really important later on. When we look at the bonds, right, of course, all these bonds in here, right, all of these are covalent bonds. And covalent bonds, of course, are very strong. But the bonds in the middle here are hydrogen bonds. The bonds that hold a base pair together, though, it holds the A to the T, or the G to the C. Those are hydrogen bonds. And they're, a 20th to a 25th of the strength of a covalent bond. So they're much, much weaker. And if you think about our mechanism, right, our semi-conservative mechanism for replication, that should make sense, which is what this question down here is asking. We don't want to break covalent bonds. We want to keep that strand intact because that is the code, right? That's the recipe for you or whatever the organism is. And so it has to be, kept intact. You want to minimize damage to that as much as possible. So strong bonds holding all that together. But to copy it, we need to be able to open it up. And so what holds the two strands together are very weak bonds. And that makes it really easy to unzip it and get access to that code, both for replication and also, you'll see later, for when we go to make proteins. I'm not sure if you remember the word denaturing. I expect you probably do. There's a video that I showed you where I drank some vinegary eggs with sriracha, I believe. But do you remember what it means to denature a protein? And I expect you do. It means we're losing its three-dimensional shape, right? And we talked about a few different ways that could be done. The acid was one, but also you probably remember temperature. Do you remember what kinds of bonds were broken when a protein is denatured? Remember that proteins in its three-dimensional shape, they have a lot of different kinds of bonds involved, right? There were covalent bonds, there was hydrogen bonds, there are ionic interactions, and then there were hydrophobic interactions. All four of those things we talked about. Take a minute, and if you need to play that part that I just said, play it again. Figure out, see if you remember which ones are disrupted when we go to denature a protein, disrupted by heat or disrupted by acid. Take a minute and do that. So hopefully you remember hydrogen bonds, the hydrophobic interactions, which are really London dispersion forces, but hydrophobic interactions is fine for biology. And then the ionic interactions, all of those can be disrupted by heat. The covalent bonds are not generally broken by heat. It takes a great deal of energy to break covalent bonds. So by that logic, when we denature DNA, how does its shape change? Because we do say this. We say you're going to denature the DNA, and we're going to disrupt those same bonds, those hydrogen bonds, hydrophobic interactions, or ionic interactions. Of course, in DNA, the only one of those that we have is hydrogen bonding. So what would be an easy way to make this happen, to disrupt those hydrogen bonds? Well, we can do it artificially just by adding heat, right? If you warm up DNA, it will denature, which means it will unwind and unzip and to make single strands of DNA. Now that happens at a much higher temperature than body temperature, but we have some technologies that we use that process. We use this ability to denature with heat to accomplish a variety of different things. And we'll talk about that later. When we look at base pairing in DNA, we have purines and pyrimidines, and you don't really need to know which ones are which. You really probably don't even need to know the words purine and pyrimidine for AP. But you probably already do, and if you don't, well, maybe now you will. But the purines, I meant to move these pictures, I'm sorry. The purines have a double ring system, so these two over here are, I'm not writing anymore, there we go. These two are purines, and these two are pyrimidines. You may have heard some mnemonics for how to remember which ones are which. Like I said, it doesn't really matter. You don't have to memorize them. But probably my favorite is that they say King Tut and Cleopatra lived in the pyramids. There's so much wrong with that. First of all, neither one of them were buried in pyramids, and you don't live in pyramids. You bury people in pyramids. But anyway, Egypt, there's a theme. Okay, close enough. The way I learned it is the ones that have a Y are the ones that have a Y. Cytosine and thymine both have a Y. Pyrimidine has a Y. That's which ones they are. So every time you're going to get one purine binding with one pyrimidine, and that's really about the space between the two sides. If you had two purines there, right, they'd be great big molecules and it would make a bulge in the DNA. Or if you had two pyrimidines, they would be too close together and that would bring the two sides in together. It would disrupt the shape of the double helix. So with one of each, they are always the same distance apart. So they form these hydrogen bonds, and guanine and cytosine form 3 hydrogen bonds, and thymine and adenine form just 2 hydrogen bonds. That becomes relevant occasionally in AP questions. So you can easily tell, I mean, it's easy to see which one is more easily disrupted, the adenine and thymine bond or the guanine and cytosine. And of course, the adenine and thymine is only 2 hydrogen bonds, so it's weaker than the 3 hydrogen bonds of cytosine and guanine. Cool. So how about this one? Think of an organism that lives in hot springs. How might its ratio of A and T pairs compare to its G and C pairs. How might that ratio be different for them than it is for, say, us or like a goat or a goldfish? So hopefully you said that you're going to get more G and C pairs in hot springs because they live at temperatures that can be near boiling. And so that makes it more likely for the DNA to denature. But if they have more hydrogen bonds holding those strands together, that makes them stronger. And that's actually exactly what we see with organisms that live in water that's near boiling. They tend to have a lot more G's and C's in their genome than the A's and T's. And it helps to stabilize their DNA at those high temperatures. So we got a bunch of different enzymes we're going to look at for the process of DNA replication. But first, let's get an idea. So we keep showing you this unwinding and unzipping, and it makes it sound like we're just taking a whole strand of DNA and just going zoop all the way down and pulling it apart. That's not the way it works, right? That would take way too long. So what actually happens is a whole bunch of these proteins are involved, and it starts unzipping in a whole bunch of different places on each chromosome. So if I've got a chromosome, there's going to be dozens, if not hundreds of places on that chromosome that are called origins of replication. And that each of those places, the enzymes will bind and start to unzip and unwind going both directions from that spot. And so you get these little replication bubbles, which is what these are here. And so as this guy builds this light blue strand here, it eventually joins up with that one, and you end up with two complete, again, semi-conservatively generated strands of DNA. So there was an origin of replication at each of these three locations. So you got these three replication bubbles. So this is a replication bubble. Each of these is a replication fork, just so you know where we are and when we're referring to these things. So let's look closely at a replication fork. We've got a few different enzymes here. So this guy here, this is topoisomerase. And you don't really need to know the names of these guys, but it is kind of nice to know what the proteins have to do to make this happen. I don't think you're ever going to get asked, what is the name of this? They don't ask vocabulary questions anymore. So that's topoisomerase, and its job is to untwist that helix. So it unwinds it, and it actually does that by breaking one of the strands and letting it swivel so that it doesn't all bundle up. So it breaks one of the strands and then sticks it back together again. It does that repeatedly down the molecule. This guy here, this is helicase. And helicase is the enzyme that unzips the DNA. You've probably heard the pickup line. I'm not going to repeat it here. Just Google helicase pickup line. You'll find it. These guys here are single-stranded binding proteins. So, or single-strand binding proteins. When you unzip the DNA, it wants to go back together again. So you have to have proteins there to kind of keep it apart. This is all, I don't really show this, but all of this is all actually attached together. All these proteins are all attached together in one big protein complex. And they all do their jobs stuck together. This guy is an enzyme called primase, and primase's job is it assembles an RNA primer and it sticks it to the DNA, because our polymerases that build DNA, they have to have a complementary strand, and they have to have a 3' end to extend. They can only extend a 3' end. So you have to stick a little primer on there. So they stick this little primer on there. The primer is made of RNA, not DNA. So it's stuck on there. And of course, since we unzip this DNA, we have one strand that has a 3' end and one strand that has a 5' end, because the DNA is, again, antiparallel. So we're going to build a new strand, right, that has a 5' end and a 3' end in the opposite direction of each of these. So this is the 3' end, so this is the 5' end of that. We're going to be building this direction off of here, and we're going to be building this direction off of here, because, again, when we have nucleic acid polymerases, so things that build DNA, things that build RNA, these are proteins, these enzymes, they can only build from the 5' end to the 3' end. What they do is they extend the 3' end. They stick another nucleotide on and another nucleotide. And that's the only thing they could do, right? These are proteins. Proteins can do one job, and that comes entirely from their shape. So the primase sticks these primers on here, and they're going to have base pairing rules here, right? So an A goes with the T and a G goes with the C. So primase drops off these primers. Oh, there it is. I apologize for that. It's legacy. What are you going to do? It's Kim. Anyway, as the helicase, right? So the topoisomerase moves down the line, unwinding, untwisting that DNA. The helicase follows, unzipping the DNA. Now I've got room to bring in the DNA polymerase, and that's these guys. And they are going to, again, extend that 3' end. So remember, this is the 5' end. I don't know why this got way out here. It's supposed to be down here. This is the 3' end. I guess I'm going to build to that. So same thing up there. So it can extend the 3' end off of that primer. Now notice they're going in opposite directions from each other. This one, the top one, is going no problem following the helicase. And this guy is actually, like I said, this is all attached together. But this guy is actually going to do this. It's just going to keep on building. As long as the helicase moves this way, this polymerase can keep on building. So this is called the leading strand, because it is allowed to build just continuously. Now this guy has a problem. The bottom one has a problem because as it unzips more and more, we can't build this part, right? That part, this enzyme can only go this direction from here. So it's only going to extend here, and it can't get this. So I'm going to need primase again. I'm going to need to stick another primer on here when the helicase unzips more of this, and you'll see that. While we're here talking about DNA polymerase, let's take a minute to think about the energy. The energy for this, right? Because we're building a polymer. And of course, when we're building, this question you should be able to answer in a heartbeat. Answer it quickly. Good. Hopefully you said that's an endergonic reaction, because you have to put energy in to build things, right? If we were breaking it down, of course, it would be exergonic, but we're building something, we have to put energy in. So where's this energy coming from? Where does the energy usually come from in a cell? You should be able to answer that. Hopefully you all just shouted ATP at your computer very loudly. But if you look closely at this ATP, so I got my 3 phosphates. We always said there's 3 phosphates, but there's a sugar here. And then there's this other guy here, that's the A for the ATP. This is adenosine triphosphate. And you say adenosine because it's a nucleoside, this thing here. It's not a nucleotide, but it's awfully close to one. In fact, right, it's a modified nucleotide. This is an adenine nucleotide. Usually the one we use is with ribose, so it's RNA, but you can make it with deoxyribose and it works the exact same way. It's got, right, these 3 phosphates, and that breaks off that third one. You break off that third phosphate, and that's how we get energy from most of our chemical reactions in our cells, or at least the ones that require energy. There you go. The boom. So can we get energy? It's a good question the penguin's asking. Can we get energy from ATP in a different way? We can also take 2 phosphates off of that. And we get energy out of that too, because that's an energetic bond right there. That's an energetic bond right there. It doesn't matter. You're getting energy from this. And so this now, that's a nucleotide. That's adenine. That's a phosphate, a sugar, and a nitrogenous base. That is a monomer of a nucleic acid. And if this is deoxyribose here, then we've got a monomer of DNA, and we can make DNA with this. And it came with its own energy. See? Are there other energy nucleotides besides just the ATP? Of course there are. We've got GTP. We've got TTP. Does the same thing. And we've got CTP. So that's all 4 of our nucleotides, right? And of course we have the same thing for RNA, and we have uracil. Works exactly the same way, but they basically come with their own energy. So what does a nucleic acid polymerase like DNA polymerase, what does it need if it's going to put another nucleotide into this spot? And what it needs is, it needs a 3' end, right? It needs a base that it can base pair with, and it needs energy. And the energy, of course, comes with the nucleotide. So this one needs a nucleotide that has adenine. And there's my DNA polymerase 3. It's going to break these 2 phosphates off of here and get the energy out. There's my energy. These sounds are terrible. And I can just keep doing that. And I'm going to flip through this quickly because it's very noisy and annoying. So this is the guy we've been talking about, this DNA polymerase, and it's doing just that. It's extending the 3' end by adding nucleotides. And as we move along, right, we extend, again, here's our leading strand, doing just great, continuing to grow all the way down the line. But this guy, again, like I said, we needed another primer and we need another DNA polymerase to extend off of here. So we end up building it on this lagging strand. This one's called the lagging strand. We end up building this in fragments. And those fragments are called Okazaki fragments, named after the guy who discovered them. So we're going to build more and more Okazaki fragments on this lagging strand, and we're going to continue to extend that 3' on the leading strand. And so we're going to have to deal with all this RNA and all of these breaks. Every place here, there's a little break where we didn't get to stick a nucleotide on a 3' end because there's a 5' end on the other side, and the enzyme doesn't do that. The enzyme only attaches to the 3' end. It can't attach onto a 5'. You need another enzyme to do that, and we'll get to them. So as these get extended, we end up with this little gap on each one of these, and we have to fix that. And like I said before, we've got to take care of this RNA. And so we have an enzyme, a different DNA polymerase. So this guy right here, as I said before, this guy right here is DNA polymerase 3. So we need another enzyme, and that enzyme is going to be DNA polymerase 1. And DNA polymerase 1, got this guy. It's got exonuclease function. So exonuclease is a word that we will talk about again. It'll come up from time to time. So we have two different things, exonucleases and endonucleases. Exonucleases can digest nucleic acids from the 5' end, and they just gobble it up going down the chain. Endonucleases digest nucleic acids just in the middle, inside, endo, within, right? So they cut random spots or sometimes very specific spots in a strand of DNA. And we'll talk more about those later too. So DNA polymerase 1 has exonuclease function. So it can attack this 5' end here and digest this RNA. So it's going to attack here and digest this RNA. And then, right, it can extend, because it is a polymerase, I just told you with DNA polymerase 1, it can extend that 3' and replace that RNA that it just digested with more DNA nucleotides. Now we still have a little gap here that we need to take care of because we don't have a way to attach a 5' to a 3' that isn't a free nucleotide, right? These enzymes only work with free nucleotides. We have another enzyme, and that enzyme is called ligase. And the word root lig means to tie or to bind. And so it just ties those two things together. And now we've got a solid piece of DNA. So this is a picture kind of showing all of that. I've got an origin of replication here where it begins to unzip. I've got a replication fork going to the right, a replication fork going to the left. Each has its own helicase, its own topoisomerase, its own sets of all the rest of the enzymes. And each is going to have a lagging strand and a leading strand. And if you look, the leading strand on this fork is actually the lagging strand on this one because they're going in opposite directions, so that should only make sense. So if you are at the very end of a chromosome, the DNA polymerase is going to go right off of this one, no problem, go all the way to the end, finish that extending that 3' until it runs out of base pairs to base pair width, and it's at the end. But this other strand, we're going to digest off that RNA, but there isn't enough room here to put a primer. A primer isn't going to fit on there. So we've got this little dangly bit of single-stranded DNA. And what's going to happen to that is it's just going to be chopped off and digested. It's gone, right? Your cells don't like single-stranded DNA. They are very aggressive against single-stranded DNA. It's because of viruses. Viruses are often single-stranded DNA. So being aggressive against single-stranded DNA, that's a helpful thing. So the chromosome just got a little tiny bit shorter. It's going to be more than, I wrote 7 or 8 base pairs here. It's going to be a lot more than that. It'll be a few dozen, right? Sometimes as many as 50 or so, or more. And so each time you go through S phase, one end of the chromosome gets a little shorter. And so as this gets shorter and shorter, eventually it gets down to the point where it's going to be interfering with genes that you need to use. And we did talk a little bit about this when we talked about cancer. So you'll stop doing cell division. And so we have these telomeres, which are just repeats. We have an enzyme whose job it is to extend these telomeres. See, as you get older, the telomeres get shorter and shorter. My telomeres are shorter, and that's why My cells aren't dividing as rapidly, and so I'm aging. I don't know if you've noticed any of the clues, but they're there. When you're very young or when you're making sperm or eggs, you have telomerase active. And we talked about how, yeah, that's kind of like biological immortality. It prevents you from aging, but it is also one of the mutations that has to happen for cancer to occur. So it's not necessarily something you would really want happening in all your cells. So this telomerase is an enzyme that just adds this 6 nucleotide repeat over and over again to the ends of the telomeres, so that when you lose that little bit, you're not losing anything important in each replication. So these get extended in every generation, basically. So this is just a slide showing kind of everybody that's involved. Okazaki fragments, primase, single-stranded binding proteins, helicase. It doesn't have topoisomerase, but it would be right here. That's fine. You don't really need to know the names of the enzymes. You do need to understand the process. You need to understand leading strand and lagging strand, understand why Okazaki fragments have to be made. And it's all about the direction, remember. So we can only extend the 3' end. Remember, proteins have a shape, and they do their job based on their shape. That's Oreo. You haven't met Oreo yet, but it's just about feeding time. So Oreo's going to meet you. So we're going to call it quits there. That's a good place to stop. That's ending the basics of DNA replication. There's a lot of enzymes in there. Like I said, you don't need to know the names, but you absolutely have to have this process locked up in your head. Like, they will ask about this. Even in the teeny tiny AP test you're going to get, I'm sure they're going to ask you about this. I hope everything's going well with you guys. See ya. Don't just wander through the house meowing. You know which one feeds you. Go to that human. Sorry.